Field, kind of going to the mammalian visual system. We have our right visual field, which is to the right of us as we're viewing, and the left visual field is on the other side. And there's a little region of overlap between the two visual fields. It's thanks to our binocular vision. And then we saw that the optic nerve passes into the brain, crosses at the optic chiasm, projects back to the lateral geniculate nucleus and the thalamus, and then we have optic radiations that go back to primary visual cortex or striate cortex, because it's striated in nature if we stain it, and the surrounding cortex is called secondary visual cortex or extrastriate cortex. And the primary visual cortex is also sometimes called V1, and the visual association cortex is sometimes called V2 through as many as V8, I think. But, typically, something other than V1 is visual association cortex. So let's look at the lateral geniculate nucleus a little bit more in depth. It's composed of 6 layers of cells or neurons. And layers 1, 4, and 6 receive input from the contralateral eye. So we have 2 lateral geniculate nuclei, one on either side, because we have our thalamus as a bilateral structure. So 1, 4, and 6, layers 1, 4, and 6 of LGN receive input from the eye on the other side, and 2, 3, and 5 receive input from the eye on the same side. There are 3 different types of layers here: the magnocellular layers, parvocellular layers, and koniocellular layers. And these are super important for kind of separating out the different visual subsystems and directing the visual information to the appropriate processing centers. So the magnocellular layers are the inner layers 1 and 2. They transmit information necessary for perceiving form, movement, depth, and small differences in brightness that gets transmitted back to V1. The outer 4 layers, so that is layers 3, 4, 5, and 6, constitute the parvocellular layers, and these transmit information necessary for perception of color, predominantly red and green, and fine details. That goes to different parts of V1. And then we have the koniocellular layers, which are sublayers of neurons in the dorsal lateral geniculate nucleus that are just ventral to each of the magnocellular and parvocellular layers. And the koniocellular layers process information from blue cones, the short wavelength cones, to the primary visual cortex. So our ability to perceive blue or anything related to blue is thanks to the koniocellular layers. This is just a close-up of the different layers, and the koniocellular layers are these kind of sublayers just to the ventral surface of each of the individual layers of the magnocellular and parvocellular layers. The inner layers are 1 and 2, that's magnocellular, and then the outer 4 layers are the parvocellular region. So the one zone of visual cortex receives input from only the parvocellular layer, one zone receives input from only the magnocellular layer, and one zone receives input from both layers. And so these 3 outputs or from the LGB, lateral geniculate body, and inputs to the primary visual cortex form the basis for 3 corticovisual systems specialized for perception of shape, motion and depth, and color. And so the processing really depends on what layer of the lateral geniculate body the projection comes from, and the destination in the visual cortex, the primary visual cortex. This table just kind of lays out some of the properties of the magnocellular, parvocellular, and koniocellular divisions, and deals more with color, Color is more a function of the parvocellular and koniocellular layers. Magnocellular layer is very sensitive to contrast, whereas the other two layers aren't. The fine detail is more a feature of the parvocellular layer as opposed to the other two. And then the temporal resolution, or the response, is very fast for the magnocellular layer and slower and more sustained for the parvocellular and koniocellular layers. We have two streams of processing from the visual cortex, and we'll talk more about those in a little bit, but we're talking about a dorsal stream that projects kind of from the occipital cortex up to the parietal cortex. And this dorsal stream receives mostly magnocellular input to detect movement and light and dark contrast. And it turns out that this dorsal stream is very important for our ability to process information regarding where something is located in the external world. The so-called where system is the dorsal stream. The ventral stream is known as the what stream. The ventral stream is important in our ability to identify objects, for example, and this ventral stream also receives about equal input from the magnocellular, parvocellular, and koniocellular systems. So those collectively, those three layers kind of come together in the ventral stream to help us eventually identify objects, tell the difference between objects, and things like that. So as I mentioned, other visual pathways project to the superior colliculus. There's also the accessory optic nucleus and the pretectum and the supraoptic nucleus. Those are kind of important for helping to set our diurnal rhythms. These latter three in particular are involved in helping to set our daily rhythms. Projections to the superior colliculus can reach the cortex through relays in the lateral posterior pulvinar of the thalamus. So that's another nucleus of the thalamus that sends projections to the superior colliculus, allowing it to reach the cortex. And that's just again, kind of the circadian rhythms, as I mentioned before, is a function of the pineal body and the supraoptic nucleus. So taking striate cortex in a little bit more detail, there are 6 layers of neurons in the striate cortex, and they're arranged in bands parallel to the surface, kind of as you see here. And the neurons respond to specific features or contours in this primary visual cortex, or striate cortex. The visual cortex combines information from several ganglion cells and allows the detection of features that are larger than the receptive field of an individual receptor. So there's a lot of convergence of a lot of sensory receptors through the lateral geniculate body on back to the primary visual cortex to allow detection of features larger than the receptive field of any individual sensory cell. Here we have the cytochrome oxidase, or CO blobs. These blobs in the striate cortex process information from the color-sensitive ganglion cells. And they're found in the 2nd and 3rd layers primarily, and faintly in the 5th and 6th layers. And the neurons in interblob regions of V1, or primary visual cortex, project the pale and thick stripes of V2. And so you can kind of see a little bit of that going on, these blobs over here. And down here are actually more of the blobs. These are the thick and the thin stripes up here. The blobs are down at the bottom. Maybe a little bit hard to see, especially if you're in the back of the room. The extrastriate or visual association cortex combines information for perceiving objects and visual scenes, and is specialized to respond to features like orientation, movement, spatial frequency, retinal disparity, and color. The structures include V2 through V5, although we'll talk about a V8 in a second. And they're specialized to respond to discrete features as above, and they're arranged hierarchically. There are two pathways in the extrastriate cortex, as I mentioned before, the dorsal stream and the ventral stream. And this what and where system was identified by Ungerleider and Mishkin, Leslie Ungerleider and Mortimer Mishkin, back in 1982. It was kind of exciting. I was in college at the time, and this was a big deal. The dorsal stream, or the where stream, terminates in the posterior parietal cortex. And so because we know that the parietal cortex is associated with not only our perception, sensory perception, but also kind of our ability to localize things in the environment, the dorsal stream enables us to perceive the spatial location of objects and motion in the external world. The ventral stream is the what system, and that system projects down to ventrally into the inferior temporal cortex that allows us to perceive form and identify objects. This is again a little diagram showing the striate cortex and projecting into the extrastriate cortex, with the dorsal stream projecting up to the posterior parietal cortex, and then the ventral stream going down to the inferior temporal cortex. So that's sort of the second level of the visual association cortex. And this is just another diagram of the what and the where system. Just a different visualization of it. Color vision is especially interesting, and trying to explain why we see and how we see different colors is quite interesting. The trichromatic color theory was proposed by Newton, Isaac Newton, and initially he proposed that white light is composed of 7 fundamental colors that cannot themselves be resolved into other colors. He actually proposed 7 receptors and brain pathways for color, and it was actually Thomas Young and Hermann von Helmholtz who came up with the trichromatic color theory, not Newton. But Young and then later von Helmholtz said that just because any color can be reproduced by mixing different quantities of red, green, and blue, just 3 colors, the color perception must be due to the nature of the visual mechanism rather than to the nature of the light itself. And so although von Helmholtz kind of amplified this finding of Young, it wasn't until von Békessy in the mid-20th century where he was able to actually confirm this idea. And so the photoreceptors in our eye are consistent with this theory. Now there's a different theory of color processing called the opponent process theory of color perception. And in this opponent process theory, primary colors are yellow, blue, red, and green. There's 4 colors, not 3. So, because we can see yellow, how do we see yellow? And this would explain how we see yellow. This kind of dated back to Ewald Hering in 1878, and is trying to explain color vision in terms of opposing neural processes, so that the experience of yellow would be maybe a more intense stimulation of a neuron, and the experience of blue might be the opposing process, less of a firing of that neuron. So complementary colors cancel each other out. They produce gray or white if we mix complementary colors together. The way that the retinal ganglion cells operate, that would be actually consistent with the opponent process theory. Trichromatic theory can explain why yellow is perceivable and why it's included in this group. Yet we can still see yellow, so maybe it's the opponent process theory that's the best account. This is just a color circle, and if you look at colors 180 degrees opposite from each other, if you actually mix equal amounts of those, they will cancel each other out, producing a neutral gray. This is kind of an example of the opponent process, so if you stare at that for about 20 seconds and stare right at that dot in the middle, then when I go to the next slide, and if you don't deviate your eyes from the image, you'll see an after image which should be a red, white, and blue American flag. And it absorbs all the color after effect. Interestingly, this has been used by butchers for years, where they decorate the inside of a meat case with parsley or other greenery, which will make the meat actually look redder than it is because of the opponent process idea. We also have to consider how light and pigment are mixed together, because they actually operate differently. An object appears red because it reflects long wavelength or red light, and it absorbs all other wavelengths. And if we mix light, we're actually adding wavelengths together to the stimulus. So light mixing is additive. If we're mixing different colors of lights, one wavelength is being added to the other wavelength. If we mix paint together, interestingly, more wavelengths are absorbed. And so in this sense, pigment mixing is subtractive. We're mixing paints together. That's a subtractive process, very different from mixing light together. So mixing equal amounts of all wavelengths of light produces white light, but mixing equal amounts of paint colors will produce black, because each added pigment absorbs additional wavelengths of light until there's sort of a total absorption of all possible wavelengths, or blackness. And that's just showing the additive versus subtractive concept with mixing lights versus paints. So kind of reconciling the trichromatic and the opponent process theory, Hurwicz and Jameson basically found in the mid-1950s that both of these theories are correct. And there are three types of color receptors at the level of the photoreceptors, so that's where trichromatic theory operates. Then at the level of the retinal ganglion cells, the three-color code gets translated into an opponent process color system. So here, neurons respond to pairs of primary colors, with red opposing green and blue opposing yellow. And there's two kinds of color-sensitive ganglion cells in the retina, red-green and yellow-blue. So it's really the photoreceptors that are more trichromatic and the retinal ganglion cells that are operating according to the opponent process theory. So nature is pretty clever combining these two notions. And these are just different receptive fields of color-sensitive ganglion cells. In this case, if yellow light shines on the center of this cell, it's going to result in the experience of yellow. And if blue shines on the surround, then it's going to decrease the firing. So when a portion of the receptive field is illuminated with the color shown, the cell's rate of firing increases. When a portion is illuminated with complementary color, the rate of firing decreases. And finally, just before we finish up for today, I wanted to talk a little bit about color blindness. This is an interesting genetic defect in color vision, and it results in the anomalies of one or more of three types of cones. And it's an X-linked type of condition, genetic condition. It accounts for, it's present in about 8 to 10% of men and about 0.5% of women. I am colorblind. There are three major types, or there's some sort of subcategories, but we have protanopia, which is a red-green color blindness. These people have a red cone peak response that actually shifted more toward the green response. So they don't really see red that much. They are liable to confuse red and green. A protan dichromat has red cones that are absent. Deuteranopia, or a deuteranope, would also confuse red and green. The green cones appear to be filled with red opsins, so the curve of response for the green cones is actually responding more to red. The deutan dichromat has no green cones, and they just have red and blue cones, but that's again pretty rare. Even rarer is something called trichromatic. It occurs in about 1 out of every 10,000 people. It's a faulty gene, not on the X chromosome. They tend to see the world in greens and reds and don't have any blue cones. So this is the normal cone sensitivity curve, and you can see that green and red, the frequencies of light are very close to each other, whereas the blue is further down. It's much less, or much shorter wavelength compared to green and red. And in deuteranomaly, where green is shifted toward red, that's kind of the reason why those people would confuse green and red. I think this is the type that I have. The deutan dichromat has no green cones, only red and blue cones. Again, pretty rare. They wouldn't see any green at all. The protanope, protanomalous people have their red shifted toward green. So that's again why they would confuse red and green. And the protan dichromats, it doesn't have any red cones, they only have the blue and the green. They wouldn't see red at all. These are kind of called Ishihara color plates, and you should be able to see a number, if you're not colorblind, in the center of each of these dots. So I can tell you I see a 25 here. Yeah. I don't see anything here. 45. Okay. Yeah, I couldn't even imagine that. This one, I don't see anything. 6. This looks like maybe a 15 or something. Or?
It's 29. So that one's green background and red numbers.
Okay. Yeah. That's a 56. That's a trick one. That's designed to fake people out if they're trying to fake color blindness. So everybody should be able to see 56. And then this one, I don't see anything.
So you can see the 56, is what you're saying?
Uh-huh.
I think the background is more blue there, and then the foreground is very much red. So that's how you can distinguish those easier.
Yeah. Yeah. Everybody sees the 56 there? Yeah. Yeah, everybody's supposed to see the 56, so if you say, no, I don't see anything, then gotcha. So these are the actual numbers that are supposed to be there. So yes, I am colorblind, so I missed all the ones that I was supposed to be able to see something. This is another one, I don't know what I see, 8? Yeah.
Oh, maybe it's a 5. I think it's a 5.
I can see the 5 and 2.
Yeah, I could maybe make out a 5 there, but it's not at all clear. And then there's a variety of other optical illusions not associated with color. This is more associated with form, but some kind of interesting optical illusions. This is the wife mother-in-law image where